Okay, the next tutorial in the sequence-based similarity module is going to be the WebLogo tutorial. In the WebLogo tutorial, I'm going to use all of the information that I have collected up until this point in my lab notebook. And I'm going to use that information to make a visual of the alignment of sequence-based similarity. You can see some of the work that I've went through and completed up until this time. Changing fonts and messing with things a little bit, I suppose. So I move on down, you can see I have a sequence, set of sequences that I've selected from the tea coffee tutorial. And as I continue, you can see the multiple sequence alignment. I'm going to need this multiple sequence alignment to create my web logo. And so I get to the bottom and you can see here's the web logo site. There's a new version, WebLogo 3, and the tutorial talks about, I would like to move on to WebLogo 3. Let's just go right there and click on Create Your Own Logo. So I'm going to need to paste in the sequence right here, get some information from that. So I'm looking for an M. I know that I can go too fast here. S, L. I don't want to go so fast because I've misplaced myself before. Like that. Yeah. Oh, I don't want that to happen either. What did I just do? Goodness gracious. Let's go back to the bottom and try again. Here we go again. Looks like that's it right there. Copy that sequence. Go over to WebLogo here. Paste it in. Keeping all the settings as default. I'm going to create a logo. Alright, this is the piece of information that we've been struggling to make all along. I would like to remind you that at the beginning of all proteins starts with an ATG codon in DNA, AUG in messenger RNA, and that represents the amino acid methionine, sort of the universal starting amino acid. So you can see that since all proteins begin this way, all, not just this one, the M is very large. Then we have a series of amino acids that don't seem to line up, and there's almost nothing there, so you don't see much. Now when you look at these single letters, the larger the letter, the more conserved the amino acid at that location in the protein. And when you see smaller letters and groups of letters, that represents a non-conserved region. So this capital F is a very highly conserved amino acid, as well as this G and this T. And look at how big that F. So this F, K, R, P, D, E, P, D, these amino acids are highly conserved in this protein, this class of proteins. I'm going to copy this image and bring this image over into my lab notebook. And you can see, try to paste that in here. And it doesn't work. And so this is a glitch. And I've figured out a workaround for that glitch. And it's pretty odd. The workaround for the glitch is do the same experiment in the web browser called Safari. Copy the image. Go back to Firefox. Paste it and it works. And you can save it. It's just really bizarre. Content exceeds maximum size. This is fabulous. Alright, how do I make the content not exceed maximum size? Let's go back. PNG. There we go. Sorry. I didn't have my default set, so I should be able to do that right here. Copy image. Go back to here, and I should be able to save it. Successfully saved. There we go. And so what are we going to write about this? Well, the beginning of this protein looks a little bit odd because there's a capital M and there's a big nothing otherwise. So this appears to be a standard WebLogo protein alignment exception of the first 10 or acids. All right, I'm going to call that the end of the WebLogo tutorial.